Hello, everyone, and welcome to today's live broadcast, Cancer Chemotherapy and the Tools Needed to Advance Treatment in a Medical Diverse Population, presented by Jennie Williams. She is an associate professor at Stony Brook University with a primary appointment in the Department of Family, Population, and Preventive Medicine and a secondary appointment in the Department of Medicine. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Jennie Williams. I will now turn the presentation over to her.
Thank you very much for the opportunity to talk to you today. My research interest is in the area of cancer chemoprevention and cancer chemotherapy, with an emphasis on the epigenetic and genetic differences that is associated with racial health disparity. Today, what I'm going to do is give you an overview of my initial research ventures, how I actually got into cancer racial health disparity through the back door of cancer chemoprevention. I'll give you some really general background information, statistics and challenges associated with racial health disparity, and I'll end this by talking about results, research results, specifically research models and omic analysis that I'm interested in working on. So to begin, our group is interested in studying factors associated with initiation and progression of a number of cancers, but predominantly colon cancer. Colon cancer is a great model for prevention, as the time from benign to metastasis can be anywhere from 10 to 15 years. Therefore, intervention can result in increased survival. Our early studies involve looking at chemopreventive and chemotherapeutic agents as far as efficacy, safety, and mode of action. So, an early observation noted that aspirin and other NSAIDs prevented colorectal cancer and adenomas. This is also true for esophageal, gastric, breast, and pancreatic cancer. The problem with these agents was that the efficiency was less than 50% and the side effects was extensive. Therefore, our group, wanting to enhance the properties of NSAIDs but decrease the side effects, developed these derivatives. We have a number of derivatives that we study in the lab. This is just an example of a couple of them. They're basically NSAIDs attached to a spacer which is linked to an active group. So without going through any of the experiments, I'm basically going to give you the facts of what we found. And these facts are that these derivatives, the efficacy is higher than their parent compound. The effect is on cell kinetics and apoptosis. They basically increase cell proliferation and induce apoptosis. They're highly effective and safe in animal models, and they exert their effects on several signaling factors. Specifically, what I was interested in and was investigating was NF-kappa B and beta-catenin and P53. However, the problem that I faced when I did my study, and this problem that is faced by a lot of other scientists, is that these initial studies were conducted in Caucasian American cancer cell lines, leaving out a huge population of African Americans and Hispanics. What is the problems or what are the limitations of the studies that are being done now? Is the availability of racially diverse tissues for research purposes, the lack of the existence of African-American colorectal cancer cell lines, PDXs, and organoids. PDXs are patient-derived xenografts, and we'll talk about them a little bit later as well. So we need these models in order to assess in vitro studies as well as in vivo studies. Okay, so to give you a background of how I got into this research, I initially received my funding by a KO1 award under the CURE program, which is under the umbrella of the Center to Reduce Cancer Racial Health Disparity. Even though I wasn't doing racial health disparity at the time, I attended mandatory meetings or workshops that actually included individuals that were working in cancer racial health disparity. Through these interactions and through just going to the meetings, I was able to get a grasp of what was needed in the area of cancer racial health disparity, and I was able to actually define a project that included cancer racial health disparity as well as looking at chemotherapeutics and looking at genetic differences. So an accepted fact is that African Americans have the highest death rates from all cancer sites combined, and from malignancies of the lung, colon, rectum, breast, prostate, and the cervix of all racial groups in the United States. We know that the burden of cancer is more acute for racial and ethnic minorities. In vivo and vitro assessments of chemotherapeutic agents and clinical trials lack rigor in respect to diversity. We also know that that differential racial and ethnic populations respond differently to chemotherapeutic agents. So in general, minorities are in poor health. They experience more significant problems accessing care. They're more likely to be uninsured. They often receive lower quality health care than other Americans. So what are the causes of these inequities? Some of them are culture bias, are assumed to be culture bias, miscommunication, lack of trust, and socioeconomic disparities, specifically barriers to high-quality cancer prevention and lacking early detection. So there are correlations that we have noted in incidence and mortality from colorectal cancer. And these correlations are age, sex, race, and ethnicity. 90% of the new cases and 94% of deaths occur in individuals 50 and older. Differences are likely between males and females as far as gender-related differences affects the incidence and mortality rates between men and women, 30 to 40% higher for men than for women. And as I stated before, colorectal cancer is highest in African American men and women compared to any other race, and this incidence rates could be 20% higher and mortality rates 45% higher. So interestingly, in New York, the state of New York, the incident rate for African Americans are higher than any other group. However, the mortality rate is not, well, the incident rate is higher, not significant. However, the rate of mortality is higher but highly significant compared to any other race. If you look at Suffolk County, or just a small area of Long Island, you will see that the disparity differs from region to region within a small region. For example, it is easier, well, it's not easier, but it's the rate of colon cancer is better for females as compared to males living in Suffolk County, Stony Brook. But this is inverse when you go to Miller's Place, which is right down the street. Overall, the incident rate for colorectal cancer in Suffolk County is not significant between any of the races. But when you look at the mortality rate, it increases for African Americans, both females and males. This slide only reiterate what I've been saying before, which is that the incident rate of colorectal cancer is highest for African American male and female. So is mortality compared to other racial groups. And it's interesting to note that this disparity is even higher, is even lower for other economic distressed individuals, such as Hispanics or Asians or any other racial groups or ethnic groups. Overall, there's a 30% to 50% higher rate of disease-specific mortality after diagnosis among African Americans in relationship to Caucasians. Even though great progress has been made in understanding tumor biology and response to treatment amongst non-Hispanic Caucasians, comparable knowledge for African Americans and Hispanic population is scarce. I'm not sure if this was a focus, but this is a study that demonstrated that after factoring for treatment modality and stage at time of treatment, the disease overall survival at 5 years and 10 years post-surgery observed in African American patients with colorectal cancer may not be attributed to tumor stage or diagnosis or treatment, but may be due to differences in other biological or genetic characteristics. And this is between African American patients and Caucasian patients. So in respect, how do we address racial health disparity? Our group is doing it through two methods. One is education and one is research. Education, we need to increase recruitment and retention of underrepresented and minority students in STEM, thereby building a diverse population of scientists and clinical investigators. Therefore, we will have diverse thinking and diverse study choices. In respect to research, we must incorporate models that span diverse populations and use these models to address biological differences such as response to chemotherapeutic agents, differences in genetic and epigenetics, and as well as looking at things like diet and what our profile of our microbiota is in the gut. And using this model, we can lead us to develop biomarkers that we can use for early detection, therapeutic targets, and prediction of pharmacological responses. So what do we know? What we do know is that African American patients with colorectal cancer have unique polymorphisms in the P53 tumor suppressor, which are linked to advanced colorectal cancer. This is at an allele called allele 72, and it's usually an arginine, but in African Americans, there's a propensity to have a protein in place of the arginine. There are more frequent high microsatellite instability, and there are distinct mutations in mismatched repair genes. So how does this affect me, and how does it play into what I'm studying? P53 and mismatch repair, as well as microsatellite instability, mediate the effects of chemotherapeutic and chemopreventive agents and differ between African Americans and Caucasian Americans in ways that are critical to their function. And compared to other populations, African Americans respond differently to chemopreventive agents. Specifically, the chemopreventive effect of NSAIDs have been shown to be population dependent. So, one other factor that plays a role in racial health disparity is that African Americans with colorectal cancer have the predominance of right-sided proximal tumors. So what does this mean? Basically, when you have a colonoscopy, it's easy to go into, you go through the anus and travel along the site of the colon. Here, this is the right side of the colon. It is difficult sometimes it's difficult to actually catch tumors that develop on this side of the colon. And when you have a sigmoidal, which is sometimes prescribed, this results in not being able to, sigmoidal actually ends around in this area. So if you have a sigmoidal, you actually don't get to see what is occurring at this end. So the general consensus is that the contribution to disparity is socioeconomic status and access to poor health. However, we do know that after you adjust for these determinants, disparity often persists. Therefore, other underlying factors such as gene regulation is involved. So this is actually a model in which we are trying to basically give a simplistic answer to what's going on. And basically, what we are trying to define here is that there are factors such as diet and environment, which is influenced by socioeconomic status, that changes the microbiota, the microbes that are in your gut, that result in increase in epigenetics and increase in genetic differences, so that you are more prone to go toward progression or initiation into colorectal cancer. So these changes in genetic and epigenetics actually result in oncogenes being increased and tumor suppressors being decreased. So oncogenes and tumor suppressors are what is driving the colorectal cancer. So some associated factors to colorectal cancer is epigenetic and genetic differences or changes which are inherent or could be acquired. And it could be acquired through diet or through the environment or through the profile of the microbiota, which is what our model is trying to suggest. So in our lab, what we are doing is we are using food frequency questionnaires, and we are looking at micronutrient levels in the serum to assess diet and diet differences between races and ethnic groups. We're also looking at environmental and social differences, specifically using the metadata from their charts in order to assess things like diabetes, obesity, age, sex, tumor location, and stage, just to name a few. And we're also doing studies where we're looking at the profile of the microbiome, and we're doing this through DNA sequencing of the 16S ribosome. A few years ago, there was a study that compared the diets of African American and Africans from South Africa. South Africa has a lower rate of colon cancer than African Americans. Here, the diet was switched between the groups, resulting in a reciprocal change in mucosal biomarkers of cancer risk and in aspects of the microbiota and metabolome known to affect cancer risk. Therefore, this study basically shows that one factor or one huge factor is the differences in diet, which we know exists between ethnic and racial groups. So our research approach is to use retrospective and prospective tissues. Retrospective are tissues that have been collected years ago, fixed and stored. Prospective tissue are tissues that we acquire as surgery is being done. So we are collecting tissues from African American, Caucasians, and Hispanics. We isolate the RNA, DNA, and protein, and we do omic analysis on them. RNA-seq, looking at the whole RNA genome. Near-seq, looking at microRNA, and looking at DNA methylation. We look at proteomics, looking at the protein profile, and exon sequencing, looking at those RNAs that are expressed. And we're looking at 16S RNA sequencing specifically for the microbiota. So as I said before, our limitation is not having tissues to do these studies. And so for one of our processes is to actually develop our own tissues. But the problem that I had initially setting up this project is that I live in Suffolk County, and the population in Suffolk County is predominantly Caucasian. This slide shows about 89%, but I think it's more like 95%. So I had to create collaborations with other individuals outside of Stony Brook area, this has allowed me to amass a number of tissues, some frozen and fresh, and some formalin fixed, paraffin embedded, which are tissues that have been fixed. So you can do later studies with them. My main source of tissue acquisition is the Cooperative Human Tissue Network, which is a part of NIH, and Downstate, which is a part of the SUNY system, SUNY being Stony State University in New York. Okay, so we have already gone over this, basically the prospective tissues I make cell lines out of, I make PDXs and organoids, and I do the omic analysis. So I'm going to go over some of the prospective tissue first, and then we'll go over the omics. So we collect metadata for our samples, and these include, but they're more than just the ones listed here, sex, age, and stage. This way, we are able to collect our samples and organize our study in a way that we are comparing apples to apples and oranges to oranges. So the first cell line that we developed are established for our study were xenografts that were tissue that was obtained from an African-American female, 69 years old. We acquired it from the Cooperative Human Tissue Network, so we called it TNR6. The cell here in this figure, what you see is that the cell lines that we generated were tumorigenic in mice. And they had the morphological phenotype of colonic cells. And this is an image just showing how we have kept them growing in culture, and this has been growing in culture for more than 3 years now. So they're well established. Since the creation or generation of CHTNO6, we have established 2 additional African-American cell lines, SB501 and SB521. This slide basically shows the H&E staining, showing morphology of the cells. TME staining basically shows the internal organization of the cells, and karyotyping is used to actually look at the chromosome numbers. So we also interrogate by characterizing our cells, we interrogate the usual suspects, those genes that are involved in cancer progression, initiation, and progression. So we've looked at a number of different proteins and compared them to HT29, which is a Caucasian cell line. This image is basically the same as before, except that this is, instead of a Western blot analysis, this is a immunofluorescence, looking at basically the same culprits involved in colon cancer initiation and progression. I would like to point out here right now the P53. If you look across here, if you look across, you would see that the P53 itself, the protein, accumulates in the cytoplasm, in the nucleus. This is indication of mutation within the P53 when it accumulates like this in the nucleus. So one of the things that we're interested in is P53 because P53 affects chemopreventive agents from being effective, as well as being a part of racial health disparity. And in fact, when we look at CHT-NO6 by sequencing, we found that it had P53 pro mutation at the allele 72 I mentioned earlier. So because we want to know how these cells behave, act when we treat them with chemotherapeutic agents, we did a panel of agents. This was just a representation of just a few that we looked at. And in most cases, the African-American cell lines were basically less effective. Well, the agents were less effective against the African-American cell lines than they were for the Caucasian cell lines. Here, we're using two commercially available Caucasian cell lines. The only exception here that we saw was for Solendac, which actually isn't one of the compounds that we formulated, but we saw that with CHT-NO6, and not the others, but just with CHT-NO6, there was Solendac itself was more effective than it was against the HT29 or the HCT116 cell. Also, what was important for us to see was that 5-fluorouracil, which is a FDA-approved drug for colon cancer, had no effect whatsoever against our African-American cell lines at the highest concentration examined. In fact, 5-fluorouracil had a stimulating effect when you looked at when you did this in vivo in an animal model. So you can see here that 5-fluorouracil stimulated tumor growth. So the other thing that we are doing is creating organoids, and organoids are actually important new models that have come out within the last few years, and they're very important for assessing chemotherapeutic agents because they actually have more of an architecture that is similar to the tissue, to the tumor itself, than having cell lines. So we went to Johns Hopkins University and was trained in order to grow these organoids. This is an image of our lab own organoid. We've only done one so far. It took us quite a bit of time to actually get all the reagents together. It's an intricate process, but once you get everything together, you can actually start flowing with your process, with generating organoids, which is now where we are. We have gotten everything organized to the point where we can actually grow them continually in our lab. So to date, we have established 3 African American cell lines. We have 5 Caucasian American PDXs, 3 African American PDXs, 3 Hispanic PDXs, and 1 colon organoid. And we use something called short tandem repeats. It's an assay in order to test to see whether or not we have any genetic variation between our tissue and the last passage of our cell lines, PDXs or organoids. The PDXs, patient-derived xenografts, they're basically they're generated from tissue discard. We basically take a small chunk of the tissue and actually insert it under the skin of immune mice. And within a month, they develop into PDXs or xenografts. And these xenografts are actually architecturally more closely associated with the human tissue or the human tumor than the xenografts that we make from cells. So the advantages of over cell lines are disadvantages over cell lines. Is cost. It is more cost effective to use cell lines. It's more cost effective to use PDXs than organoids. The organoids and the PDX have more realistic microanatomy, so you actually seeing what's going on basically real time, or are realistically closer related than the cell lines themselves. They more accurately predict the PDXs and organoids more accurately predict the efficacy of chemotherapeutic agents. So our basic goal is to generate a database that would link the same sample set to the omic analysis, proteomics, RNA-seq, DNA methylation, and look at microbiota, the diet, and using metadata to assess what differences exist on a socioeconomic status basis, and also using in vitro and in vivo models that we are generating in the lab to actually assess these omic analysis in a clinical setting, preclinical setting. So what we're doing right now is we're doing RNA-seq and exome-seq using a core facility at Cold Spring Harbor Laboratory. We're also looking at DNA methylation using Cold Spring Harbor Laboratories as well. And we're doing microbiota from a research consortium in Colorado that is collaborating with us. So the genomic analysis, I'm moving just a step further, just to give you an overview of some of the stuff that we have found and associated with RNA, microRNA, and methylation as it compares to African Americans and Caucasians. So before we start, I would just like to say that microRNAs are non-coding RNAs. They basically they're specifically regulatory RNAs. They're processed as as a large unit and transported out of the nucleus into the cytoplasm, where they're processed to the point where they become mature microRNAs. So these mature microRNAs bind to the 3' untranslated region of the messenger RNA, and depending on how tightly they bind, they can repress translation, or they can send the microRNA for degradation. So the first thing we did was look at microRNA differences using microRNA microarray. This first slide here just shows the difference between tumor versus parent adjacent normal colon tissue. We saw something like 49 upregulated and 40 downregulated microRNAs when you look at fold changes of tumor to normal. When we compared African Americans to Caucasians, we were able to find 5 microRNAs that had a race or tumor effect. And by doing quantitative RT-PCR, we are actually able to validate 3 of these microRNAs. One of them of interest being MIR-182 and MIR-183. So MIR-183 and 182 are oncomirs, and they belong to a family of transcription factors, or transcription microRNAs, regulatory microRNAs, and they the downstream targets of MIR-182 and 183 are FOXO1 and FOXO3. FOXO1 and FOXO3 are suppressive proteins, and we were able to show an inverse correlation between the upregulation of MIR-182 and 182 and the downregulation of FOXO1 and FOXO3. The differences were seen to be significant when you looked at normal versus tumor. The differences, there were slight differences when you looked at African Americans compared to Caucasians, but these were not considered to be significant. We also looked at proteomics. This is mass proteomics, in which we looked at differences between normal and tumor, African American and Caucasians, and we compared them together and outside, looking at African Americans to African Americans, normal and tumor, and Caucasians to Caucasians, normal and tumor, and then compared those factors, Caucasians and African Americans. What is interesting, what we are trying to now dig more deeply into are the differences that we see in the tumor of the African American and the Caucasian. We also looked at gene regulation using RNA-seq, and this actually just shows the differences with colorectal cancer tumor and adjacent normal, a number that were upregulated and numbers that were downregulated. We also looked at the differences between African American Caucasians and looked at those that were upregulated and those that were downregulated. Okay. This is just a slide that shows different genes that were upregulated and different genes that were downregulated. We actually are still assessing what all of this means. We have like a slew of genes, and we're just trying to assess what role they play in cancer initiation as well as racial health disparity. So we also looked at methylation. So as you know, genes can be controlled by upstream methylation at the promoter region. So we compared this in African Americans and Caucasians as well. And this slide basically shows the total methylation and those that are hypomethylated and those that are hypomethylated in African Americans that had colorectal cancer and in Caucasians that had colorectal cancer. So from that slide, we were able to deduce that African Americans had 1,588 hypomethylated and 100 hypomethylated differentially methylated regions. And in Caucasians, it was 109 hypomethylated and 4 hypomethylated DMRs. This represents a 14.6-fold and a 25-fold difference, respectively. So what we are trying to assess now is what does this mean? A lot of the hypomethylations were not necessarily in promoter region. So it's interesting to see that there is this much of a difference in methylation, even if it isn't in a gene or associated with the promoter of a gene. So also what was interesting is that one of the genes that were differentially methylated between African Americans tumor versus adjacent normal was CHIL1, which is a tumor suppressor. And this gene is also actually downregulated by MIR-182, which kind of shows that it's being silenced by methylation as well as the upregulation of MIR-182 may also affect its being expressed. So what we have found is that we have a microRNA profile that denotes 89 differentially expressed microRNA, normal versus tumor. We have validated significant differences in the expression level of 5 microRNAs between Caucasian Americans and African Americans by quantitative PCR with significant main effects of race and tumor. We also have performed immunohistochemistry analysis of samples showing an inverse correlation between the expression of MIR-182 and the level of 2 of its predictive target proteins, FOXO1 and FOXO3A. We've shown unique polymorphism. I didn't discuss this, but we have shown unique polymorphism in P53 tumor suppressors of African Americans at allele 72. And specifically, we've done this with one of our cell lines, which has a pro-pro instead of an ODGPRO phenotype. We have differentially methylated profiles generated by Illumina tumor versus normal, as well as African Americans versus Caucasian. We have generated and characterized 3 African American cell lines, 3 Hispanic cell lines, 3 African Americans, PDXs, I'm sorry, 3 Hispanic, 3 African Americans, and 5 Caucasian PDXs. This will provide us with models to assess chemo-response, chemoprevention, and chemotherapy. So overall, the goal of our research is to acquire genetic, epigenetic, and molecular understanding of cancer and provide details that may assist in the development of chemopreventive agents that will span the risk population. I would like to emphasize that our research is not exclusive, but is inclusive. We're interested in what creates cancer, what causes cancer to progress, and in all races and all ethnicities. There are a number of people who I need to thank who have been great collaborators. They're at Stony Brook University. They're at Moffitt Cancer Center. They're at Washington University and SUNY Downstate, as well as Cold Spring Harbor and the University of Colorado. I thank you for your attention.
Thank you, Dr. Williams, for that informative presentation. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen and click on the send button. Dr. Williams will be following up with all of your questions via email. I would also like to thank LabRoots. I would also like to once again thank Dr. Jennie Williams for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.